Thank you for all coming. This is a streaming video of Biomedical Engineering Institute's Tuesday seminar series. It is jointly organized by Life Science and Technologies, Center for Life Sciences and Technologies, or BU Lifesci of Boğaziçi University, also supported by İSEK, Istanbul Health Industry Cluster. So there will be hopefully lots of students, graduate students, but also other faculty members from our institute or Center for Life Sciences, from Boğaziçi University, as well as from other universities. And there might be also participation from companies, startups, or other interested parties from our mailing list. Today, we will have a special guest, Pınar Özbay. She received her undergraduate degree in chemical engineering from Boğaziçi University. Later, she completed her master's degree in computational science and engineering at Technical University of Munich, and finished her PhD from ATH, Switzerland. She, starting with his PhD work, she worked on brain anatomy and function with novel MRI techniques. She joined the advanced MRI section of Laboratory of Function and Molecular Imaging at NIH, Bethesda, working on multi-modal acquisitions in sleep and wakefulness. Works with various modalities, behavioral, physiological, fMRI, EEG data, as well as MRI. It will be a great pleasure to hear her talk. Thank you for visiting us. She is also actually physically visiting Istanbul. So if anybody wants to meet her, talk to her, maybe get a separate appointment from her as well. It will be also helpful for some of the Boğaziçi faculties, I presume. Thank you very much, Pınar Hocam. The floor is all yours. Thank you.
Thanks a lot for this nice introduction. It's a great pleasure for me to be here and having a chance to present you my work, even it is virtually. So before moving to my research experience so far, I want to tell you a little bit more on my academic background, but Cengiz Hoca explained it really nicely, so I won't really repeat it a lot. But I graduated from Boğaziçi University in 2009, and then I wanted to have a more interdisciplinary experience, so I received my master's in computational science and engineering from Technical University of Munich, and there I had a chance to work at GE Global Research, and it was located in our campus, and there I first time had an experience in the field of MRI. And later I wanted to continue my interest to a PhD degree and moved to Zurich, and there spent 5 years in a collaborative project together with ETH Zurich and University Hospital Zurich. And that was under the topic of quantitative susceptibility mapping. And today I will try to describe you its methodology and progress and applications to the human brain. Following my PhD, I moved to the USA, and since then I've been working on more multi-modal acquisitions and analysis during sleep and wakefulness, and it's been almost 4 years, and I will today also discuss with you some of my projects I've been involved. So as said, I'm really excited to present you my research experience so far. It will have my flow of the talk will have 2 parts. Initially, I will describe you my PhD projects mostly, and then move to my postdoctoral research more on multimodal acquisitions. So to start with, I would like to first describe what is magnetic susceptibility. So it is a quantitative dimensionless proportionality constant that indicates the degree of magnetization of a material or the tissue and its tendency to interact and distort an applied magnetic field, in our case, our MR scanners. So on the right, we see a famous diagram from a paper by John Sheng in 1996. So here on this susceptibility spectrum, what we see are that there are three main parts. So there are diamagnetic materials, paramagnetic, and ferromagnetic. And the part that is shaded is the region called Mri compatibility. And what we see here, for example, water or soft tissue has a diamagnetic property as it has a negative susceptibility, while, for example, magnesium and aluminum is on the positive side. So if we look a little bit close, what we are interested in MRI, what we call indeed diamagnetic, it's a diamagnetism relative to soft tissue, for example, calcifications. And on the other hand, for example, deoxygenated blood or liver with iron overload, we call them paramagnetic, but relative to the soft tissue. So why are we interested in mapping that susceptibility distribution, for example, in the human brain? So we have several main susceptibility sources, such as I list on the left, like tissue iron based on ferritin, and that makes QSM highly interesting and applicable to visualization of like deep gray matter structures. Also, clinical cases such as Alzheimer's disease or like tumor lesion detection for patients with multiple sclerosis. Also, QSM is highly sensitive to measuring and visualization of venography, microvillids, and that makes it also applicable to functional studies. Other susceptibility sources are like myelin, calcifications, inflammations, and they open a wide range of applications as well in the brain, such as imaging tumors or, as said, MS or Parkinson's disease cases. So here is an example that I wanted to show you from literature. So in this case, those are images from an elderly volunteer showing numerous hypointense spots in thalamus, shown with, for example, solid arrows. Also, other dark spots shown within and around ventricles together with veins. So in a typical T2 star weighted image that we acquire from MRI, is not able to differentiate, for example, iron deposits from calcifications. On the other hand, the below image is a slice from QSM susceptibility mapping, and it is revealing hypointense calcifications. So those are the little black dots, and it can differentiate those from the iron deposits occurring in thalamus. So what we see bright on a susceptibility map typically shows us a paramagnetic property relative to soft tissue, and a darker spot means a diamagnetic property relative to soft tissue, such as calcifications in this case. So when we say susceptibility in the field of MRI, that also reminds us a source of artifact. And the reason is that there are local field inhomogeneities originate partly from susceptibility differences between tissues, what we are interested in, but there are also artifacts may be occurring due to abrupt changes in the susceptibility between, for example, around air cavities or in changes between like bone to tissue regions. So now I will try to make the link between magnetic susceptibility and MR data, MR phase data particularly, that we are interested in. So each image voxel here we see on the phase image can be approximated as a magnetic dipole, which produces a dipole field extending beyond the voxel. So indeed, a signal intensity from a voxel here is not a local tissue property. It is extending beyond itself. And those dipole fields originating from different voxels follow a superposition rule, and it is resulting from a convolution relationship between the field and the susceptibility distribution. This is a representation equation of the phase in relation to the magnetic field and the susceptibility distribution in image space. And indeed, that can be expressed in a more easily in like a multiplication in K-space, which is the Fourier domain, Fourier transform of the measured image. So what I would like to describe you a little bit more here, we have, if we are trying to measure the susceptibility distribution of the brain, we have to go from the phase image to the susceptibility, and hence that involves an inverse problem. So that D term here goes to the denominator of the phase, and that creates an ill-post inverse problem because the coefficients of this equation become 0 at a magic angle. So before trying to solve the inverse problem, an easier approach would be to understand the forward field calculation. To perform that and also to optimize various parameters in the pipeline, we conducted a simulation study based on human torso model. So on left, we see a human torso model filled with known susceptibility values, and outside is filled with air. So this is chi is the susceptibility distribution. This is again showing the equation between phase and susceptibility distribution. And if we want to calculate the phase differences, like here, then that equation becomes straightforward because it's a forward model and we are just convolving with the dipole kernel. And then we can extract our brain, and this would be similar to what we get from an MR acquisition, a phase image. But of course, that the reverse approach won't be that easy. So how to measure MR phase? So in this work, we conducted a study with a phantom filled with gadolinium solutions. Here you can see different tubes filled with different solutions. And also, we also performed a study on healthy volunteers. This is data acquired at 7 Tesla during my PhD studies. And we can see from the magnitude images that there is a nice contrast between gray and white matter, and also in the ventricles, typically we observe in a T2 star weighted image. And below, we see the phase image all acquired, basically calculated from exactly the same complex data. And this is showing like the dipole pattern as we discussed before, since the shape is like circular. And this is the phase image of exact same slice from the same data. So what I want to also stress here a bit that it's not typical to save the phase data in an acquisition. So one most important thing is that we have to save the phase data, either making it available from the MRI console, or if possible, of course, we can save the raw data or complex data and then calculate our images afterwards. So now I will try to give you a bit recipe of how to calculate susceptibility mapping. So that includes multiple steps that I won't be able to go in detail today, but briefly, we are acquiring, let's say we have the phase image from our scanner, and that can be a single echo or a multi-echo that needs to be combined. As this is a phase data, it has restrictions from -pi to pi, so we have to phase unwrap the data. And later, we also have to remove the background field that are due to mainly those air tissue interfaces I showed you before. So that can be performed with various methods. And once we solve those two steps, say that we have a field distribution which is more local now from our brain, then the next step would be the inverse problem. So if we try to solve the inverse problem directly, we wouldn't be able to have a meaningful image at all. So what we need to do, either we can use like a threshold-based method in case space, or we can perform iterative least square algorithms. Here I give you just some examples from the literature, which are more like established algorithms. So in this particular work, I use an iterative least square solver. And here is a slice from the susceptibility map. And the voxel intensity is linearly proportional to the underlying tissue magnetic susceptibility. For example, in deep gray matter structures or in large veins, we see that it is brighter versus the soft tissue. And if we compare it to the exact same slide from a T2 star weighted image, we can clearly see that there are like different and indeed complementary information, which makes QSM really applicable to various applications. So before moving to applications to the brain, I want to show you briefly a project that I was involved, mostly in indeed hardware development. So in T2 star-weighted imaging, the images can be strongly suffer from artifacts caused by spatiotemporal field fluctuations, and those can be induced by, for example, deep breathing or arm movement. And they cannot be fully corrected by post-acquisition data processing methods. So a method has been introduced which minimizes such fluctuations by means of magnetic field monitoring with NMR field probes and real-time full up to third order field stabilization. So this hardware has been developed at ETH by the MR Tech group, and then this is also like a spin-off company, Scope. It's called Scope, and now they made the hardware indeed open to everyone. So as example, deep breathing, as said, creates those field fluctuations that these are now, each color represents a field fluctuation acquired by each probe. So that, of course, degrades our images as well tremendously. So here, I want to just show you briefly an illustration of this hardware system because it has multiple components. So here we have the NMR signal from the field probes, which is sampled by a standalone spectrometer. And the field information then is fed to a controller actuating the gradient and shim amplifiers. And the aim is actually to correct for deviations from the desired field configuration. So this is the same experiment I showed you in the previous slide, that we asked volunteers to take deep breaths during the scan. And on top, we see the field evaluation from the probes without using the field control system. And here we see the corrected field changes across all probes, which work pretty well. So as said, that affects, of course, our image quality. And then here is an exemplary slice from QSM acquired from 7 tests from a healthy subject. And what we see here, indeed, we are not even able to differentiate boundaries between gray matter structures. Also, there are highly distorted areas due to ghosting artifacts and blurring. So I will now try to flip back and forth to show you the improvement with this system, which makes everything more visible, the boundaries of the structures, the deep gray matter structures, and also it is reducing the all blurring and ghosting artifacts. So that such hardware improvements and developments, of course, are highly also important for applications to clinical cases, such as subjects with high body mass index or patients with neurological diseases, because those subjects are known to have irregular breathing, for example. So now I will give you a brief overall overview on the projects that I was involved during my PhD, as I was also interested in applying QSM to the brain for studying physiology and functional studies. So I have like 3 parts of projects, namely oxygenation, clinical, and functional. So the first study I want to show, in this work, we wanted to assess oxygenation with QSM and quantify the change in oxygenation in different regions of the brain, as well as in large veins. We recruited 12 healthy volunteers, and the experiment involved inhalation of medical air and also hyperoxia, which is 100% oxygen. So first outcome of this project was QSM was less, say, susceptible to air tissue interfaces, and those long-range effects from paramagnetic oxygen in frontal cavities was more distorting our susceptibility maps, because we are indeed applying various processing steps to remove such background fields. So this is a comparison to a more traditional method called R2 star, basically 1 over T2, reverse over the T2 star images. So the results, we quantified change in susceptibilities in gray matter, in large veins called sagittal sinus, and also in ventricular regions filled with CSF. So what we observed was a large reduction in susceptibility shifts towards diamagnetism due to decrease the oxy versus oxygenated hemoglobin ratio in veins. And on the other hand, in CSF, we had an opposing trend, for example, due to paramagnetic effect caused by dissolved paramagnetic molecular oxygen in the ventricles. QSM also allowed us to quantify change of venous oxygen saturation between hyperoxia and normoxia. So we performed that with use of the following equation, and we calculated that hyperoxia induced a change of venous oxygen saturation around 10% in sagittal sinus. So such studies was important to us because we actually wanted to extend that to a clinical work. And in the next project, a small cohort of patients, we wanted to assess the tissue response in tumor regions to respiratory challenges. So in this work, the aim was to also see the response in different regions of the brain to respiratory challenges around tumorous regions. So in this work, for example, this is from one subject having a large glioblastoma in around ventricular region and anatomy scan. So I was able to acquire and analyze data from 5 patients with malignant tumors. So this study was in a large collaboration together with anesthesiology and radio-oncology and as well radiology of the University Hospital Zurich. So in this case, patients inhaled medical air as well oxygen and as well carbogen, which is a mixture of oxygen and carbon dioxide and serves as a more like a vasodilator. So here on left, we see a contrast-enhanced T1 weighted image showing the region of glioblastoma tumors inside being necrotic area as it's dark and surrounding the active tumor area and the edema. So in QSM, we were able to better delineate that active region in tumor, for example, and also differentiate regions of necrotic area and edema. So then we made a regression plot between susceptibility change and versus magnetic susceptibility, so the baseline susceptibility and during normoxia case, so the medical air. So what was really interesting here that contrast enhancing tumor regions with high baseline susceptibility, say the lesions around here, show the marked susceptibility reductions under respiratory challenges. So this was the reduction during hyperoxia, and this is reduction during the hypercapnia. So this also allowed us to differentiate, for example, even the regression plot, the edema and necrotic regions, they didn't show such a reduction under respiratory challenges. So that may allow us in future, like an early identification on hypoxic tumor areas in MRI, and even may support indeed therapy planning and monitoring, taking into account their response to those challenges. So the last part that I would like to discuss with you, using susceptibility data in functional studies. So in fMRI, in functional MRI, say we are showing our subjects in a visual task in the scanner. We are expecting that task to induce a neuronal activity and increase in blood flow, and that would increase the ratio of oxygenated versus deoxygenated blood. And the result we observe is an increase or an increased activation in our fMRI signal. And this is indeed due to a decreased susceptibility, so a shift toward diamagnetism. So here on left, we see slices from T2 star weighted image acquired at the NIH. And now that subject, we showed that subject a visual checkerboard for around 12 minutes. So here are like 4 adjacent voxels from a box shown here. And these are from raw magnitude time series. So what is interesting here, even this is really the raw signal, we already can see the fluctuation due to the task they are visualizing. If we do the same thing and we calculate the phase image and then try to visualize a pattern, temporal pattern in time domain, unfortunately, we are not able to see any task-related pattern, and the phase data is relatively noisy, and it has a drift inside and all those reps and also background fields. So we have to process our phase data and then repeat that at each time point to indeed analyze our susceptibility data similar to fMRI. So these are the time series voxels from phase data, and these are now the time series voxels from susceptibility data. Although it's still noisier with respect to magnitude data, we can clearly see that we now are able to have an information indeed in the signal related to the task activation. So once we have that our time series data clean and pre-processed, there are various ways to analyze our functional data. That can be a seed-based correlation, seed-based analysis based on basically Pearson's correlations. That can be based on also model-based approach called general linear modeling. Also, there are various approaches called data-driven methods such as independent component analysis. So in this work, acquired at 3 Tesla with 9 subjects on a group level analysis, what we see on top is a result activation map induced by a visual task in bold fMRI, so the traditional bold fMRI. And in below we see similar analysis based on susceptibility data. What was interesting here, the activations were mostly around similar regions. However, we observed consistent bilateral draining maze with a significant response to the visual paradigm. So we continued that work also at 7 Tesla, and that was also we observed that by directionality in activation maps, together matching the fMRI activity. So of course, I was not the only one observing that in QSM community. There has been also other publications discussing that issue. And then one argument was that that could be due to a counterbalancing effect of blood fractional oxygen saturation and blood volume changes. Yet to understand the mechanism behind that, this is still an ongoing research in the field that I think we need to design a more clever experiment, including maybe both traditional fMRI, as cerebral blood flow measurement can be detected by ASL, and also we can include the cerebral blood volume based fMRI, and then FQSM or susceptibility based fMRI can find its place in the world of fMRI. So other directions or interest that I have on top of functional studies is, for example, solving the inverse problem. That can be done as nowadays it's also like a rising methodology by use of deep learning, like convolutional neural networks and proper training data sets. In addition, of course, neurological applications such as Parkinson's disease or epilepsy patients would be really interesting to apply QSM. And I know that the Institute also has an interest in animal imaging. What I have described so far, all of them mostly can definitely be extended to animal research, such as studying animal models of Alzheimer's, cancers, and any pathology like more sensitive to iron and calcium. So with that, I would like to move to my projects that I'm being continuing at the NIH during the last 4 years. So just give a break. So I feel lucky to be involved in such a large overnight sleep study at the NIH, and that more or less started when I moved there indeed. So that study is really special because the subjects really spent the night in the scanner, and we are acquiring data from fMRI, physiology, and EEG. So this is a simultaneous study ongoing. And I've been involved both in the experiment part, processing, and analysis of the data. That gave me a chance to learn many other things in neuroscience indeed. So here, before subjects come to visit indeed, there's an extensive period beforehand. So that includes like internal screening, in-person screening, then they get an actigraph from us, and there is a home monitoring period, which is around 2 weeks. And then they also have also noise habituation part. And then they come for the inpatient visit. And actually overall, considering the number of people got in touch with us, the success rate of the experiments is around like 10%. So this is one run throughout the study, and then around 45 minutes, as we are collecting data throughout the night, around 7 hours, of course, with options that giving to subjects to give breaks. And here, what we see, the subject, for example, initially have a wake stage, and then going to a light sleep called non-REM 1, non-REM 2, and then even towards deep sleep called non-REM 3. During our experiments, there are cases in our control, but there are also scenarios that not in our control. For example, the mental state of our subjects. So our subjects can be sleepy instead of being awake and alert, or they can be really stressed due to the task they need to perform. And those things do affect our systemic physiology through different pathways. And we can indeed track and understand those changes better if we collect data to understand our cardiac and respiratory cycle through the experiments. So to do that, we are collecting data from respiratory bellows and also pulse oximeter attached to a fingertip. So here I want to show you what we are measuring from a PPG signal. So a PPG is an optically obtained plethysmogram that can be used to detect blood volume changes from the fingertip. It's widely available almost all in scanners, hospitals, and nowadays, unfortunately, even in our homes. So in the field of fMRI, what is PPG used for mainly is to calculate heart rate variation, beats per minute. But on the other hand, we can have other information from that signal as well. For example, here in this case, that drop in the amplitude or the envelope of the signal is indeed due to the fluctuation in finger skin sympathetic tone, and a drop meaning here is due to a peripheral vasoconstriction. So there hasn't been a lot of fMRI studies use that information. Here I want to show you 2 examples. So on the left, what they call is pulse height variability is indeed that amplitude of the PPG signal. And they are showing correlation maps between that pulse height variability with fMRI. So what is interesting here, those are data from 4 different subjects, and they had really different patterns, especially in the brain, which wasn't explained fully. And I think the reason is that all those subjects had different arousal states and mental states, and hence they had different fluctuations and covariances between fMRI and the PPG signal. And on right, in this study, authors used a similar device to a pulse oximeter, which is called functional NIRS, which is also attached to a fingertip. And in this case, they calculated independent components. And then here they are showing the spatial maps of those components derived from their fMRI data. And on top, those are the components giving highest temporal correlation with the peripheral data, which are mostly around sensory regions and large veins. And here, interestingly, they show the most negative correlation they observed with the peripheral data, which was around white matter. So in our data, particularly during light sleep, we also observed intermittent drops in our PPG signal, as seen here. This is a raw PPG signal, and those are mostly accompanied by increased heart rate variation. So this is an exemplary magnitude image that we acquired throughout the night from 3T, and this is a signal change, normalized PPG signal, and those are the drops we are extracting from this particular signal. And here I'm showing you a whole brain fMRI person signal change. And what is interesting here, the drops in the fMRI signal are indeed following the drops in the PPG amplitude. So here is a result from one subject. So PPG amplitude and fMRI correlation. So during light sleep, we observed that strong correlations across whole brain with a strong contrast between gray and white matter. So we think that this is indeed pointing to a systemic vasoconstriction, which could result as a delayed mechanism in white matter with respect to gray matter. So we tried to explain that in a model shown here. So as attributed to a vascular transit delay that during vasoconstriction can be happening in white matter, which results as indeed a contrast between gray and white matter signals. So here, what we see as a positive correlation, indeed, a signal drop in fMRI following a PPG amplitude drop. Our observation is also in line with literature here on left from our subject data. So the negative correlations we are overlaying with the magnitude data, and it is highlighting really deep white matter and ventricular regions. And here on the right, we see a different subject from another work, but showing bolus arrival of gadolinium injection, which is also coming to white matter with a delay of around 5 seconds versus gray matter. So following that work, we wanted to further understand the effects of sympathetic activity contributing to our signal, and hence we used in a follow-up work also EEG in addition to fMRI and PPG, and we analyzed data during autonomic arousals occurring in light sleep. So in literature, the relation between arousals and the PPG drops has been actually introduced. For example, here, an acoustic noise inducing an arousal, which is seen as a burst in the EEG, and then is followed by a subtle PPG amplitude drop. In our data, we also really observed a similar behavior. So those asterisks show the burst in the EEG data, and they are nicely followed by the PPG amplitude drops. So what are those bursts of activity in EEG? Just as a brief information, they are called K complexes, and they are in the signatures of subcortical arousals occurring primarily during stage 2 sleep. And they have been in literature already associated with episodes of increased sympathetic activity. So we included 7 subjects with segments of the light sleep in the following work, and I used a freely available toolbox called Sleep Toolbox to automatically detect those K complexes across subjects, and overall detected around 200 Ks with a 90% match with PPG amplitude. So let's check a bit the temporal changes of our signals. On top, I show person fMRI signal change in gray matter and following a PPG amplitude drop, and this is the low frequency EEG power, which is a marker of K complex. So if we, for example, focus on this rectangular box, we can see that those increases in the EEG power are mostly followed by PPG amplitude drops and a drop in fMRI person signal change. So next, we can do a cross-correlation, lagged cross-correlation between those signals. And here we see that this is a low-frequency EEG power correlation with amplitude of the PPG and fMRI. So the 0 point can be regarded as the events of K complexes. So what that plot tells us, following a K complex, we are overall in the subjects having a PPG amplitude drop with a lag of 6 seconds, which is also later followed by a fMRI signal drop. And of course, if we cross-correlate those 2 signals, we have a positive correlation. So that temporal information was interesting, but on top of that, we also observed that special pattern between gray and white matter between those signals. So here, I first want to tell you that I swapped the color bar here to have a better comparison between those correlation maps. So in this case, if a hot color means that a positive correlation, while here it's a negative correlation. So basically an increase in the EEG power results as a decrease in the fMRI drop. So what is interesting here from the voxel-wise correlation maps, we have also an understanding in the legged behavior. For example, they are following a similar pattern, which means that they indeed point to a common sympathetic origin. And there is also a 6-second of delay we observe here, but we also can calculate from, of course, from the temporal correlations. So that roughly 10 seconds delay we observe in the fMRI, what we think is that that cannot be due to a pure neuronal system, as a hemodynamic response is indeed more or less expected around 5-6 seconds. So this needs to be something different or in addition to neuronal mechanism, which in this case pointing to a sympathetic contribution. So if we zoom out from our light sleep, this is the spectrum I showed you before, showing the EEG spectrum, the sleep stages. What I want to show you here briefly is that this is the EEG power and the PPG amplitude and fMRI signal. So if we check the EEG power change across different sleep stages, we see those little increases, jumps in the EEG power due to the K complexes, and those become largely variable in deep sleep. Those are markers of deep sleep, which are called as slow waves. If we check our peripheral data and the fMRI signal, we also here see nicely how they covary together during light sleep, and they become really stable during deep sleep. And here is an exemplary rotation motion information also throughout that long scan. So we were also interested in the wake conditions, as of course fluctuations in sympathetic tone are not exclusive to light sleep. So we checked our wake segments from our sleep study. And of course, as expected, there is no subcortical arousals during wake conditions, and hence we don't have a pattern or correlation between EEG power and fMRI, while we have a still even less visible, but still a pattern and strong correlations with or PPG amplitude and fMRI. So later we checked another physiological measure we collected, so that we calculated respiratory volume from our respiratory bellows and cross-correlated PPG amplitude and RV. So what that plot show us, across subjects, we most of the time during wake segments, most of the time we observe a negative correlation at a negative leg. That means that subjects tend to inhale deeply, and that is followed by a PPG amplitude drop. And that mechanism can be another triggering factor of sympathetic activity. So there are various other scenarios can trigger that. And in literature, one is painful stimulation, for example, they showed. And another one is errors in mental tasks. They discussed and they showed a trend toward larger pupil sizes, which is another indicator of increased sympathetic tone. And lastly, years before, it has been indeed already shown that finger vasoconstriction was observed after deep inspiration. So this is a measure of volume of right third finger measured by a glass plethysmogram, and here is a deep inspiration, and the drop in the volume is following a deep inspiration. So now I will show you really briefly experiments including acute deep breathing with a continuous visual task and a cognitive task, which in this case based on an arithmetic operation, because I wanted to disentangle those contributions from more like respiratory effects, or more like a carbon dioxide effect from a pure sympathetic origin. So I collected data from respiratory bellow, pulse oximeter, and also an eye camera in this case from a 70 scanner. So the eye camera was really interesting. Here I will show you a case that where subjects indeed asked to focus on a visual task. However, as said, not all the subjects perform as we want. So indeed, an eye camera collection, data collected from an eye camera can be really useful in terms of using the data as a regressor, but also tracking the behavior of our subjects. So here is an example from the acute deep breathing data. This is a continuous task with visual checkerboard. So I asked subjects to take a deep breath whenever they see a green dot on the screen instead of a red dot. So this here, for example, one trace from raw respiratory signal where subjects subject nicely inhaled deeply, and these are followed by PPG amplitude drops. And in addition to these, we also observed pupil dilation following deep breaths. And once we checked event log fMRI signals across all subjects, so those are average across 13 subjects, we also observed a lagged behavior in fMRI and signal drops mostly occurring in around sensory regions. As said, respiratory deep inspiration is such a complicated system because it is triggering sympathetic activity, but also the effects can be more due to a local carbon dioxide effect. So that's why I also wanted to design a cognitive task, hopefully to see effects without deep inspirations. So in this case, instead of deep breathing, I show a summation on the screen, and the subjects just were asked to do the arithmetic operation in their mind. And that didn't really particularly change their respiratory signal, luckily, but we observed PPG signal drops and also pupil dilations. So this is just an example of group level task-based results. And here is just to show you that subjects really did perform the cognitive task, because those are called the arithmetic related brain regions, such as intraparietal sulcus. And at a later leg, similar to deep breathing, we also observed large signal drops in and around gray matter, particularly in sensory areas. So overall, use of these physiological and behavioral signals as regressors would be definitely interesting to understand contributions coming from more vascular origin or neuronal origin. And indeed, a refining fMRI response model based on peripheral measures would be definitely interesting as well. And on top of that, such measures and methodologies can be applied to, for example, patients with epilepsy, as they are known to likely to have breathing cardiac alterations. And as in my initial part of my talk, all those aspects, methodologies, and tasks can be also extended to animal imaging, such as one can conduct behavioral studies with animals with use of trackers, physiological, and even electrocortical measures together with fMRI. So with that, I would like to thank to my colleagues from Zurich and also Germany and my current lab at the NIH. And also thank you for your attention.
Thank you. I hope you guys can hear me. Excellent. Thank you for the nice talk. And we are on time. We have time for questions, I believe, from the audiences. I think we will also have one-on-one session afterwards with faculty members, as far as I know, or within a group today. So maybe we can ask students from participants outside Boğaziçi and students here, and see whether we have several questions. Let me see, I was also looking at this one. Okay, so there is one question from Mesut Uğur. I know he is interested with microcirculation a lot, especially hyperemia and trauma, and especially treatment of local temperature for things after trauma. So you can see his question maybe on the chat. Do I have to repeat it for you, or maybe answer it in two ways?
Okay.
In a general sense, think of all the MRI techniques that can be utilized for trauma, hyperemia, or in general, giving an information about what can be utilized in MRI technologies, and also maybe more specifically on local temperature changes or edema or trauma, how these type of factors affect the measurements that you are getting on the fMRI side, or oxygen level.
Yeah, okay. Okay, so this is more, about the trauma patients, I believe. I haven't worked with trauma patients, but in subjects during oxygenation studies, there may be also hyperemia. Is this what you mean, Mesut?
Yes.
Okay. I haven't experienced that, luckily, to be honest. But experiments we did in Zurich during oxygenation, they were mostly around 8-10 minutes, and they were all. So it was like I think under limit of creating an hyperemia in subjects.
Could you repeat that?
So our studies, our experiments was around 8 minutes during oxygenation. So I think this wasn't enough to create an hyperemia in healthy subjects. So we didn't have any problems indeed with our healthy subjects.
Okay.
Yeah.
Okay, Burak Hoca has a question. Can you answer his question from the chat?
Yeah, okay, I see now. No, I didn't include any brain connectivity analysis in particular, but I mean, in terms of connectivity, what they call connectivity is also indeed based on correlations. So all those discussions around correlation analysis can be also extended to connectivity analysis. And hence, in fMRI, there are now, for example, various discussions on white matter connectivity, functional MRI in white matter, and they discuss it to be a neuronal origin, which also could be, but I think one has to be careful and consider all those physiological changes occurring with delayed mechanisms in white matter, and maybe regress out signals properly, and then consider various aspects to say directly it's a neuronal activation in white matter.
Okay, can I just thank you for the answer? So you're saying that many of the connectivity analysis can be improved by regressing out the effects of the autonomic system, right? Is that what I understand?
Improved, I wouldn't say directly improved, but better understood. But yes,
especially fMRI, because it's related with blood oxygenation.
Exactly, yeah.
Okay, thank you.
Yeah. Can I ask a vocal question, or do I have to type it? This is Alpay.
Go ahead. Go ahead, please.
Yes,
no, thank you very much.
Afterwards, Esin Hoca will ask a question also.
Oh, I'm sorry.
It will be better to ask it verbally, because then it will be getting recorded.
Okay. No, Esin Hoca is first. She was before me, so she should go ahead. Sorry.
That's okay, Alpay Hocam. Please proceed. That's fine. I can wait.
Okay, one of you guys first. Esin Hoca, go first.
Esin, please go. Yes.
Okay, then. Pınar, thank you for this nice presentation. I was wondering about the oxygenation study in malignant brain tumors. I know that when, tumors have less oxygen and then lactate forms, radiotherapy gets affected, and actually radiotherapy is less effective on tumors with high lactate. I remember this from years back. So you also mentioned that radiation treatment planning, might benefit from your oxygenation study. I was wondering if they did that afterwards, if they followed it up and then maybe applied it in brain tumors, or not yet? No,
not yet, unfortunately, not yet, yeah. But the radio oncolog, she was highly interested in also indeed like PET MR acquisitions, and they were even planning further continuing that. But as far as I know, they didn't, at least they didn't follow up with me. Yeah.
Okay. I mean, that can also be combined with MR spectroscopy and lactate measurements. Definitely. Because there are sensitive, lactate editing techniques. Yeah. That could really benefit. A combination of those will be very interesting. Okay, yeah. Thanks.
Yeah, thank you.
All right. Pınar, thank you very much for the wonderful talk. Very illuminating, several topics. I hope we'll see more of these from you in the near future. Just out of curiosity, maybe it's a detail, but with the fMRI T2 star experiment and the, I'm going to call it fMRI QSM experiment, the change in phase is fishy in the sense you need to process it to get some information out of it. And the amount of change with the blood flow for the QSM part, at least this is just my feeling. I don't have a clear view of it, but it's not enough maybe to create something directly with all the in quotes confusion coming from the phase structure. It's difficult to detect the what T2 star emphasizes in the functional case. Any thoughts on that? You did show some results, but how do you feel about it? I don't know how in any other way to ask it, but after processing QSM, I wouldn't expect too much of a change with the susceptibility map, because whatever is there near the veins or the arteries is still there in terms of susceptibility. There's maybe little change. Any thoughts on that?
Yeah, so it wasn't definitely straightforward to process that data in a functional study, and you're right. Things are way more visible and larger in the magnitude image, and overall the person activations are also less in susceptibility-based fMRI versus magnitude-based fMRI. I mean, there have been also like literature before trying to use directly the phase data to create functional activations, and then they had really sporadic like spots in the brain, and they didn't even resemble any pattern. But once we solve the inverse problem, I believe that the information is getting more local, and that more local information, I think, also has a meaning that there may be actually less active regions, and it is not spread around the voxels as in magnitude fMRI. But on top of that, in general, to study the resolution, field strength, they all affect a lot studying susceptibility in fMRI.
Okay, thank you. Thanks.
Yeah. Any other questions? We have a few more minutes. I think the students who are eager to wait for their own lectures at 2. I know few of them are here. They can slowly, they are allowed to leave. I got their participation on file. But for the remaining people, we can maybe also start, continue with few more questions. Okay, there are a few other comments on the chat, as I can see over there. So, but no real questions. How about I can ask about the question about usually most of these studies are in very well controlled environments with normal volunteers / graduate students. When it comes to the patients, especially in pathologies of the brain, let's say complex pathologies, or let's say after trauma or stroke or even cancer, then the whole assumptions that relies on this flow, bold relationships is becoming very unpredictable, right? What has been shown so far by you or by other people in terms of seeing these oxygenation measurements in actual disease patients, for example?
So I think you're trying, what I understood is like things are not that easy when it comes to patient studies, and
yes, right. The underlying pathology, I mean,
yes,
neurovascular coupling,
so yes,
neurovascular coupling relies almost or perfectly functioning brain physiology, right? If there is a pathophysiology changes in, I don't know, a mass producing region, or after surgery, or even if after epilepsy, I would think, and the normal physiology, these coupling mechanisms are no longer predictable, right?
Yes, that is true. For example, following seizures, there are also studies showing malformations also with use of with use of QSM as well. So especially if one wants to use a relatively novel method under those patient studies, I think one still has to collect various modalities and make a comparison and make those methods complement each other, because it would be also hard to understand directly from one particular imaging method, which is definitely relatively new, because yeah.
Okay.
But overall, for me, that being involved in a patient study was, even it was not a large study, it was really important that I was able to see a real world impact. And on top of that, at the NIH, I also volunteered as like an observer in an epilepsy surgery where they implanted, where they made implants during epilepsy surgery, and following that they are like monitoring the subjects, patients, and one things become much more valuable when you see all those and like research in EEG, MRI, and the whole surgery environment can come together and help a patient. But it's also really hard, yeah.
Thank you very much.
Yeah.
So are there any questions? We are actually finished our time, but this is the time to ask questions freely. You can just don't bother typing, just freely take on the floor or talking. So in terms of the first impact on the clinical disease will be on the epilepsy, is your feeling of some of these oxygen imaging or susceptible advanced oxygen imaging work?
So yes, in general, I think susceptibility mapping, one can be patients with Parkinson's disease or MS, Alzheimer's. So all those clinical cases sensitive to iron, indeed, are highly valuable and for QSM and detect for the detection of lesions and as well. And in addition, like stroke cases or epilepsies that with seizures, following seizures, there occur malformations in the brain, and then all those detections can help for the future and the benefit for the patient, I believe.
Okay.
Sorry, can I ask a question?
Go ahead, Alireza.
Okay. Thank you for your presentation. Can you please clarify more about the time resolution of fMRI and EEG, because time resolution of MRI is very few, and for EEG the frequency is kind of high. How could you compare these data together?
Yeah, that's a really great question. Oh. Okay. That's a really great question. So EEG is really special because it has high temporal resolution, like up to milliseconds. And then on the other hand, in fMRI, the echo time, so the resolution was 2 seconds, for example. And then what eventually I need to do more averaging around those 2-second segments. And this can be like on a window base or like basic mean average, or you can get the peak. And I mean, I tried many of those approaches, they come to a common result, I have to say. But just as a comment, in fMRI literature, there has been also tremendous research on increasing the resolution temporally. So there are now works going like 0.5 as well in fMRI. And that, of course, then allows us to track very rapid changes as well, and a better comparison with fMRI and EEG.
Okay, thank you.
Yeah.
And I have another question, and I am kind of, I don't know to ask it, or do we have time or no?
I am happy to chat later as well if you want.
Okay.
If we have a timing issue.
Because every time I read these neuroscience articles, the most important question, most of the time it comes up and it just pops up, and it is very hard to answer, is the causality of the spikes or getting a very good, how to say, a peak in the spectrometer. So I want to know about the causalities of these, how to say, these images you got.
This is a huge question. So maybe a quick answer, then maybe another seminar might be warranted there.
Yeah, I'm sorry. I'm so sorry to ask this.
Everything is correlational in neuroscience. Neuroimaging most of the time, unfortunately.
Yeah, I'm sorry to ask this. Just
no, it's okay. It's a very good question, but it opens a symposium.
Yeah.
No, maybe I just wanted to know, maybe in the group you worked this, maybe you had this discussion together that what is the causal, maybe there is something else. I just wanted to know if you have discussed something like that, maybe we can know about that.
So I think I don't have a direct answer to that, but I am really happy to discuss with you later. That's a great question, but it's complicated.
On DCM.
Dynamic causal modeling, is it?
Yeah, I was giving a homework to him, but
oh my. Yeah.
Okay, excellent. I guess somebody has to pull the plug, so that will be probably myself as the backup of the scenario. I guess since Pınar Hoca is here, if you have urgent questions or personal interest in this matter on neuroimaging, especially some of these advanced topics going into the tissue and getting more and functional imaging on localized tissues, relationship with that with other type of measurements, physiological signals and EEG, please contact her. The contact information is available. She is here for a couple of days to also chat. Depending on the social distancing requirements with additional faculty members as well. For people who are interested in medical imaging, maybe Alpay Hocam, Esin Hocam, Daniela, Alberto Hocam, and myself will also hopefully take some time to speak one-on-one with Pınar Hoca while she's in town. So thank you for this excellent talk. So let's wrap this thing up on time. And looking forward to many fruitful talks and collaborations, hopefully in the future, and see